They tend to speak a little slower, you know, and sometimes they can't keep up with me, and I start talking about this science stuff. So I've lived there too long, can you tell? So we had to make a shift at Probe several years ago, and we moved to, well, if we can't get to the campus itself, we know our Christian students are having numerous problems and difficulties with the secular onslaught that was going on in the classroom. Not so much arguing against the concept of God, but just ignoring it altogether. It's irrelevant. Smart people, intelligent people don't think that way. So just you're best just leaving that back home. You're here in the university now. You want to be an educated person. They don't worry about these things. So we began doing something we called the Mind Games Conference. And that was designed for upper high school students, juniors and seniors, to help prepare them for the intellectual challenge of their faith they would face when they got to the university. And that started off very, very well. We did our first one in 1991, and eventually we had a week-long camp that we did at a campground in Arkansas that went on for 14 years. Hundreds of students went through that. Again, just communicating a Christian worldview and introducing them to the worldviews they would hear on campus. And one of my favorite things to do, which I won't do here for you, I'll spare you, is in advertising that Mind Games Conference to Christian high schools, Christian youth groups, different places in Arkansas, Texas, whatever. I would come as an imposter, if you will, and I would pretend to be a atheistic university sociology professor, and I was researching the religious beliefs of young people, and I just was there to ask them some questions. Now, very often they didn't know that I was really a Christian speaker, and so they actually thought that's what I was. And so, following after 1 Peter 3:15, which says that we should always be ready to give an answer to anyone who asks you to give a defense for the hope that you have, and do so with gentleness and respect. So that's what I did. I simply asked these high school juniors and seniors, first of all, are you a Christian? And if they said why, if they said yeah, I would say, why? Now think about that yourself. If I were to walk down here and get in front of your face and say, you're a Christian? You said yeah, and I asked you why? Why are you a Christian? Now, what students would often tell me is how they became a Christian. Well, I asked Jesus in my heart. And I said, well, man, that's just gross. What's he doing in there with all the blood and everything? That sounds awful. What do you mean, yes, Jesus in your heart? Well, no, I trusted him as my Savior. I said, oh, you trusted him. What did you trust him with? Your wealth? Did you give him your piggy bank or something? What trust? No, I trusted him with my life. Oh, how'd you do that? You gave Jesus your life? What, did you carry it around with him or something in a paper bag? You see what I did? I would just start asking questions to push. What is it you really believe? And why do you believe that? Well, you can tell it didn't take long for most of these students to just get to the point where they didn't know what to say. And what we have seen over the last 10 years particularly, the more research we've done, we began finding that these Mind Games conferences, after a very good 10-year run, we did them in churches as well as at the campground, well, we started having more and more difficulty getting churches to secure a conference and have us come. We had difficulty once we were scheduled to come, getting people to register, and we only had a minimum of 50, and we'd get 35 and 25 and 20, and churches would ask us for a break. You know, can you get? And I said, you signed a contract. It costs us just as much to send one when there's 50 people here as when there's only 25. What do you're asking us to take the loss. And so we began to reevaluate everything, and we began looking at our actual presentations. Maybe we're just boring people, but we kept getting positive feedback on how excited people were. They didn't know such information existed, and I wish more people were here. Okay, so it's not the delivery. And we thought, well, maybe it's just a marketing thing. So we had a professionally done DVD video. There was a 1-minute version, a 3-minute version, and a 7-minute version, playing different contexts of the church, and that still didn't hardly make a dent. Something's really not right here. And then we began noticing some of the research done by the Barna Group about Christian worldview. And they were researching the opinions and thoughts of Americans about the religious life. And what they did, they started this in 1997. About every 3 or 4 years, they've repeated the same study. And they, first of all, I think they have a very good definition, by the way, of what a born-again Christian is, because they ask you to identify yourself one way or the other. And they say that if you're born again, you have an ongoing relationship with Jesus Christ that's important to you still today, and you are certain you are going to heaven because Jesus died on the cross for your sins and you trusted him as your Savior. You think people, if they agree to that, they're born again? I think they're not just asking, are you born again? Yeah. Well, no, they want you to define that. And you might be surprised to believe that about 45% of American adults identify themselves as born again. Do you see that in our culture today? Does it look like 45% of American adults are born again believers? No, it doesn't. Well, they started going a step further, and they asked 6 questions to ascertain whether these born-again Christians or American adults as a whole have anything resembling what they would call a Christian worldview. Question 1 was, is there a creator God who is all-powerful and still in control of things today? Pretty basic foundational principle, right? Is there absolute moral principles, moral truth, okay? Did Jesus live a sinless life? Trying to ascertain, is he divine? Is Satan a real person or entity, not just some figure of imagination? Is salvation, can you earn your way to heaven? Okay, the answer to that's no, right? And then the last one, is the Bible accurate in all its statements and all it proclaims? 6, you would say that's very foundational, very basic, right, to a Christian worldview. Of all adults in America, with a representative survey, only 9% can answer all 6 questions accurately. When you limit it to those who claim to be born again, it doesn't get a lot better. It only goes to 19%. Now that should tell you something. That's basic, foundational. Start asking additional questions beyond that, and the number's going to shrink further and further and further. When you ask adults in America who are 18 to 23, I hope you're sitting still. And don't stand now. You might want to sit back there. When you ask adults in America who are 18 to 23 those same 6 questions, only 0.5% answer all 6 questions accurately. 1/3 of those adults claim to be born again, and the percentage only rises to 2% when you ask born again 18 to 23-year-olds. Our generation has failed in many ways. We haven't passed on to the younger generation what is true and how it ought to affect your life. So Probe is making a major change. We realized that what we were doing was addressing those who already understood, who already got it. Those are the ones who would come. They'd listen to anything we'd have to say. But we were missing 80% plus of those in our churches today. And our young people, it's 90% or more who don't really understand even the foundation of a Christian worldview. And we call those individuals who don't understand, we call them cultural captives. A second passage we've used for decades with Probe is Colossians 2:8, where Paul tells us to see to it that no one takes you captive according to philosophy and empty deception. That's according to the tradition of the world. That's according to the tradition of men, rather than according to Christ. We failed, and we have failed to pass that on to this next generation as well. I don't blame the young people. I point the finger here. So we're changing some things at Probe Ministries, and we're going after those cultural captives. How can we gain their attention? How can we communicate that Jesus' death on the cross didn't just save us from hell, he saved us to a life that is real and vibrant and far more exciting than you could ever dream. But we've allowed ourselves as a church as a whole to become captive to the world around us. And we are far more influenced by the secularization of culture than we are by the words of Scripture. Bible knowledge isn't enough. It's how does it impact here? How does it impact here? How does it impact your heart, your soul, your mind, your strength? That's how we're to love God, with all 4 of those things, right? Heart, mind, soul, strength. But if you're lagging in one of those, you're lagging in all 4. And one of the big areas where we believe there's a huge cultural captivity, and you know this, is with evolution. Evolution is assumed in most agencies of our government and most aspects of our culture, whether it's music, whether it's television, whether it's movies, the entertainment industry. Evolution is what is promoted. That is what is assumed to be true. And therefore, since we are just evolved beings, then you see what starts falling away. How can there be absolute moral truths if we're just evolved beings? Morals have to change. Oliver Wendell Holmes, the great jurist. Anybody hear that name before? Oliver Wendell Holmes? Yeah. He was a Bible-believing Christian when he went into the Civil War. But what changed was the horror he saw in the Civil War. And by the time he finished, after the Civil War, he read Darwin. And he began to realize that not only do animals and physical, biological things change over time, but so do morals. And therefore the law has to change too. Oliver Wendell Holmes was the very first jurist to say that we legislate literally from the bench. That's what we need to do because society is going to evolve, and therefore law has to evolve with it. He was the one when we had the first sterilization laws in the U.S. back in the 20s and the 30s, and when those were first challenged in the courts, mandatory sterilization for those who were deemed to be mentally handicapped or physically handicapped. We actually forcibly sterilized over 60,000 people here in America because of that. 33 states had automatic sterilization laws, and when that was challenged in the Supreme Court, it was Oliver Wendell Holmes who said, with this one particular case, he said 3 generations of imbeciles is enough. But what's the bottom root of that? It's evolution. It's evolution. So my continuing romance, if you will, with this creation-evolution discussion, first of all, goes back to 1973 as an undergraduate student, where I found a little book on a shelf in the Christian bookstore there that said Evidence Against Evolution. I was a zoology student, major, concentrating on animal ecology, and I looked at that little book, and the title said Evidence Against Evolution. I didn't know there was any. And just reading that little book just started a 30-plus year journey, if you will, which eventually led me to writing the book Natural Limits to Biological Change with Lane Lester. And again, I did bring a couple of books, but I only did that because I hope to have a brand new revised edition out within 12 to 18 months at the latest. I've got 2 more new co-authors that are working with me to help get the work done. As president of a ministry, I don't have a whole lot of time to write anymore. I don't know how some of those other guys do it, but it's pretty tough for me. And what we tried to do in Natural Limits was what does the biological data actually tell us? What is the primary current? What is the evidence we really see? And what we actually see, what I'm going to demonstrate for you, is that we see that there's limits to change. Yeah, we find evidence of change, but we keep running into limits everywhere we look and everywhere we go. And no matter how we study it, what kind of organisms we're looking at, yeah, there's change, but there's always limits to change. And I could have called it the created limits to biological change, but I wouldn't have gotten many evolutionists to read it, would I? So I called it the natural limits to biological change, hoping to gain a readership from those who might not necessarily be friendly at the same time. I even had my evolutionist professor from the University of North Texas read it for me to make sure evolution was represented accurately and fairly. He agreed that it was. He said, I think you did a good job, but I still don't agree with you. Okay, fine, but I only asked you to see if it was represented accurately. And it still is unique in that context in which we offered the neo-Darwinian perspective. I explained what it is. I used their evidence. I used their quotes, said this is what neo-Darwinism is according to them. And then we offered a critique. And then a new theory at that time, punctuate equilibrium. I defined punctuate equilibrium in a whole chapter according to them. This is what they say it is. This is how they say it's supposed to work. And now then I offered a critique of that. And then we finally had another alternative. Well, we have some new things we're going to be adding in, not just updating those, but new whole areas that weren't even a possibility when the book was first written in the mid-'80s. But what I'd like to do is walk through some of this for you tonight and just look at a few things. Now, when you look at nature itself, you begin to see a couple of things. Convergence is something talked about a lot in ecology, evolutionary discussions, that we find similar forms to accomplish similar types of adaptations. Biological change is not limitless, since certain solutions seem to be arrived at again and again. What do I mean by that? Well, placental versus marsupial mammals, for instance. There are both placental and marsupial wolves. There are placental and marsupial rat-like creatures, and they're similar forms, but they clearly have very wide divergence in terms of how their young are born and how they live their natural history. And so evolutionists would say these evolved separately, right? These have a common ancestor, these have a common ancestor. But we find the similar kinds of forms that attack and adapt in similar ways to similar kinds of environments. So something must be limiting that change. Why else would you have similar placental and marsupial mammals? There's only so many ways to make wings, right? We've got flying insects, we've got flying mammals, we've got flying birds, but there's only so many ways to do that. Wings have a lot of similarities, even insect wings to bat wings. There are similarities to those things. Fins, the ways you're going to move through water. There's only so many ways to move through water. Fins are going to have, no matter whether on a mammal, whether they're on a fish, whether they're on a crustacean, they're going to have similarities. The physical environment itself poses some kinds of limits. There's only so many ways to move across land. There's different kinds of appendages, but they all do some of the basic things, okay? So we have physical limitations somehow to these to change. So there are physical constraints to the kinds of adaptations we can have. Now we have to ask the next question, are there biological constraints? Well, we'll talk more about this later, but the stunning burst of body plans and the Cambrian explosion indicates that there's some kind of limit to change. When this came out in Time magazine in 1995, I was stunned by this statement by the evolutionary developmental biologist Rudolf Raff. What he said was, there must be limits to change. Wow, really? Why? After all, we've had these same old body plans for 500 million years. Okay, you can quibble with his timing. But he makes a very real and important point. That all these special types of body plans do seem to appear suddenly in the Cambrian period. And they don't change. Why not? There seems to be a limit being imposed here. Natural selection. This is where change is supposed to happen. All the different examples we've had of natural selection or the different models that have been proposed show us some interesting things. You've got artificial selection, you've got the peppered moth, and we'll talk about Darwin's finches as well. And I'm just going to focus on these three examples because I think that, again, they make the same point. We find some elements of change, but there are always limits to change. Artificial selection. Well, Darwin used it in Origin of Species as sort of a precursor, an example of what he meant by natural selection. When he looked at the pigeon breeders and all the different kinds of pigeons and doves that you could breed, and he said, man, if humans can do that in just a few years, just imagine what nature can do given millions of years, right? Well, it's not exactly the same thing, and probably most of you know that. We've done all kinds of artificial selection. I just threw a few examples up here: roses, dogs, pigeons, horses, cattle, protein content in corn, sugar content in beets. We have selected all of these different kinds of organisms. But you know what happens? We always run into a barrier. We run into a barrier. All the selected qualities eventually fizzle out. Even today with roses, what we end up having to do is graft things together anymore. Actually, just going through the process of selecting for different kinds of roses doesn't get you much anymore. The protein content in corn, well, you know, it kind of, you increase it, and it reaches a plateau, and it just levels off. You can't get any more protein in corn. The same thing with sugar in beets. And when we first started selecting, it goes up and up and up and up, and then it just levels off and it stops. You know, dogs and pigeons, horses, cats, we've done a lot of artificial selection, but we always end up running into a barrier. Now, chickens don't produce cylindrical eggs. We haven't seen that. You could try to select for it if you want. Might not work. Probably won't work. We can't produce a plum the size of a pea. We can make small plums, but we can only get it so small. Or a grapefruit. There are limits to how far we can go. Some people grow as tall as 7, 8 feet. Some people as small as 3. But we don't find 2-foot people, and we don't find 12-foot people. There are limits to the kinds of change you can investigate and breed for in organisms. Peppered moth has been around for a long time, and it's fallen on some tough times if you're aware of the story that's gone on over the last 10 years or so. But even 10 years ago, before a lot of that information came out, there were already a number of unsettling issues about it. It was one of the earliest examples of natural selection. In fact, it was the first example of natural selection. When was Origin of Species written? 1859. When was the peppered moth experiments and stuff done? Anybody know? 100 years later. 1950s. Our first example of natural selection. Took 100 years. Okay? What do we find out? Well, we got changes in frequency of the melanic form versus the peppered form. We had 2 different forms of moth. They're illustrated for you there, and they're 2 different types of bark, a peppered variety and a melanic form, or a dark or black form. And initially, in most of the populations in England, only the peppered form was seen for the most part. There were always black ones that showed up. If you went into different kinds of collections, you could eventually find a black melanic form, even going back into the early 1800s. But what began to happen is that with the coming of the Industrial Revolution and pollution levels rose in England, and so what happened is that the melanic form began to become more prevalent, and the peppered form became less and less observed. And what they reasoned is that bird predation was thought to be the mechanism. As you can see, if you look over all the way to the right, the peppered variety is very highly camouflaged on trees that have lichen growing on it. And if the lichen is gone in the photo below, well, now the peppered variety is very easily seen. And they did some experiments trying to demonstrate this, but the conclusion they came to was that the peppered variety was just hidden from the birds, and they came along and picked off the black ones before the pollution. And when the pollution killed all the lichens on the trees, now the peppered variety was easy for the birds to see, and they came along and ate those. Well, and so the melanic form became more prevalent in the population. That was the story. However, the actual genetics of this melanism, or this change in the color, isn't known. It wasn't known in Kettlewell's time in the 1950s. It's not even known today. Both forms of moth appear to have always existed. The mechanism of selection now is also in doubt. When they've gone to different parts of England, and even different parts of the world, North America has some peppered moth similar in New England, the consistent corollary is just pollution. Not lichens on trees, not birds, predation, none of that. Just, is there pollution present? We have no idea what is going on with that. Also, by the way, all the photos you see in the textbooks, the birds are glued onto the trees. They're never found on tree trunks where they're easily visible by birds. They're underneath the leaves in the small twigs and leaves and branches of the upper parts of the tree, and so the story is really kind of falling apart. But well, even if it was a true story, you've got 2 forms of moth always there because of a change in some aspect of the environment. The frequency of the 2 forms just switched. That's not real evolution. That's not what we need. That's a change, but there's a limit to that change, isn't there? 2 forms, always through the entire story. Just different numbers of them. Then you come to Darwin's finches and the research of Peter and Rosemary Grant. What's pictured for you over there on the far right is Daphne Major in the Galapagos Islands. It's on the north end of the larger Santa Cruz Island. They would spend 3 to 6 months on that island every year for over 20 years. And they marked and captured and followed every single Galapagos finch on that island. Every single one. So they came up with some very interesting and worthwhile data. It's one of the most exhaustive studies over a 20-plus year period of time of what happens as the environment changes. And the Galapagos Islands, they're actually very dry and arid. They're on the equator, but the waters around them are fairly cold because the waters come from the west and they come from deep and they come up, and the waters are fairly cold, 60, 65 degrees. It's not tropical, even though it's on the equator. And they go through rainy seasons, El Nino, El Niña, and there's dry seasons and wet seasons, and there are wet years in a row, there are dry years in a row. And they found something very interesting as they began doing more specific genetic studies. Bill and I spent some time at the Galapagos Islands together with ICR back in, I think it was 2005. And pictured for you here are 11 of the 13 Galapagos finches. Now, what's a little deceiving about these photos, because of the different colored backgrounds, it seems kind of easy to pick out the different finches and so forth. And in real life, it ain't so easy. It's not easy at all. In fact, this one here, the photographer, the naturalist, has large tree finch with a question mark. He's not sure that's what it is. When we were first on the islands, began seeing some of the finches, it was impossible to tell who was who. The top three finches across the top there, the small ground finch, the medium ground finch, and the large ground finch, look exactly the same except for their size. If you don't have three of them sitting on a branch next to each other, how do you tell which one you got? Right? The guide that we had, and we had the same guide for us, our group of about 12 or so, for each of the excursions we took, he made a really interesting comment referring back to the grants, and there's a Darwin Research Center there, and he said, yeah, they have kind of a running joke at the Darwin Research Center there in Santa Cruz. Only God and Peter Grant can identify Darwin's finches. Okay? Now, what they began to discover here, and I'll go into this more in detail later, is that these top 6 finches here, which are all the ground finches, the 2 cactus finches are also considered ground finches, they're probably all one species because there's interbreeding between all of them. First of all, one indication, comes from a paper where they analyzed the mitochondrial DNA of all of Darwin's finches, and on the top you see those same 6 ground finches, all clustered very, very tightly together. And where you want to draw the cluster is right here. Notice how little of degree of separation there is between all 6 of them. Literally what that says is you cannot separate these by their mitochondrial DNA. You can't do it. Even the tree finches, and there's a little bracket there for you just below the ground finches, really very similar, very, very close together. Hard to tell who's who and how much difference there really is between the different tree finches. Other data, the ground finch species interbreed at a frequency of about 5%. The grants were also able to show that the offspring of these hybrids, the hybrids themselves, were fertile with other hybrids and going back to the parental species. So they could back-cross back to the original parents. The tree finches also interbreed, although they haven't been studied as extensively, so we don't know to what degree, how much fertility. As I showed you, the ground finches cannot be separated by their mitochondrial DNA. The beak size, we know, is heritable, because they could follow that from generation to generation. But we don't have any idea, really, to this point, what the actual genetics is, and how many genes are involved, how many alleles might be involved. Bill and I were talking a little bit about that earlier, but for the most part, there's still a lot of unknown aspects of that. So there is a radiation, but it's very minor, and it's actually still in progress. And actually what they have found is that during the they have a series of wet years. Then there's a lot of seeds that are produced from the plants. You have a wide variety of seeds produced, small ones to very large ones. And actually, the beak sizes tend to contract a little bit during the rainy seasons. During long dry seasons, all the dry seeds have been eaten up. The plants are not producing seeds anymore, and the only thing left are the larger and larger seeds. So the beaks tend to get a little bit larger during the dry seasons. But it goes back and forth. Smaller beaks, larger beaks, smaller beaks, larger beaks, measured by about 1 millimeter in difference, by the way. Okay. Now, I guess to a finch it makes a difference, okay? But 1 millimeter isn't going to do much for me. Now, the examples of the peppered moth, Darwin's finches, well, they're good evidence of natural selection acting on the variation that's already there in a natural population. And no one, as far as I know, no creationist that I've ever met argues about natural selection. Natural selection is real. Everybody understands that's an actual process. But what does it really tell us about evolution? With Darwin's finches, it's just going up and down, up and down, up. There's no direction to the evolution. With peppered moth, you've got a dark form and you've got a peppered form, and that's it. And they shift a little bit back and forth, but there's no real evolutionary change. So again, what we're seeing is there's evidence of change, but there's limits. That's what the evidence is telling us again. But that's where mutation is supposed to come in and save the day, right? You have to have new sources of variation. Natural selection is not sufficient. The near-Darwinian synthesis, mutations of all sorts fill that role. Excuse me. Apparently that must be me. So I will turn it off. There we go. All right. Now, these mutations actually fall into 3 main categories, as we know today. Initially, when I did this lecture, there were just 2. The first 2 I mentioned. Mutations to structural genes are those genes that actually code for proteins. Okay? Or mutations that are to developmental genes, genes that are involved in the early embryological development of the organism. But now we also have a new area that's rapidly rising as even far more important than the actual mutations that could happen in these actual genes coding for proteins. And those are in non-protein coding RNAs. And I'll describe that a little bit more as we get a little further down the road. You may have heard the term junk DNA. It's been used when I was in graduate school. It was used repeatedly, quite regularly. They already knew that only 2% of the human genome actually codes for proteins. What's the other 90% there for? They couldn't figure it out, couldn't see any reason for it. They found lots of extraneous sequences, lots of repetitive sequences that didn't seem to be able to do anything. They didn't code for anything. Didn't seem to be made into RNA at the time. So they just must be junk. Things that they look like viruses, some of them, endogenous retroviruses and all kinds of different things. They just kind of randomly spread themselves through the genome. Just a bunch of junk. Well, now they've discovered, with all these genome projects and looking more concretely at the entire sequence of the genome, is that almost all of your genome, almost all of your chromosomes, is transcribed at least into RNA. Now, only about 2% of that ends up being translated into protein. But almost the whole genome is transcribed into RNA of some form. Now, in a cell, that means it's got to be doing something. And that's a huge, huge discovery over these last 10 years in particular. They had no idea that this junk DNA was useful for anything. Now they're finding out it's actually got very, very critical functions in the cell. Now, structural mutations, those that are, let's see, we've got different kinds of functions to your liver, for instance, has proteins that are unique to the liver for the operation, the function of the liver. Those would be called structural genes. Your enzymes, all the enzymes in your body, those would be called structural proteins or structural genes. Okay. Now, I'll define structural genes as those which code for a protein that performs a maintenance, support, specialized function. Developmental genes influence specific tasks in embryological development. Most examples of mutations that we've run across, that all evolutionists talk about, involve microevolutionary changes with structural genes. So most of the mutations you've ever heard about are structural gene mutations, and it's all microevolution. Okay? Antibiotic resistance, for instance. There are already proteins in the bacterial cell which, if the antibiotic comes in, these proteins pump it out. Well, you can have a mutation to that structural protein that it just pumps out faster. You haven't really changed anything. It just works, apparently. Excuse me. Not sure what that's doing here, but we will. No, I ain't going to answer it. It's one of those text rings, you know. Antibiotic is deactivated, or it's just deactivated in the cell. That's already happening in most bacterial cells, but they just do it faster. Target molecule inside the cell is modified so it's no longer sensitive. A lot of these antibiotics affected the ribosome, the translation machinery of the bacterium. Well, and it attacks one certain aspect of it, and if you mutate the ribosome, well, it's no longer sensitive to that antibiotic anymore, but you haven't really changed anything. These mechanisms of resistance can be achieved naturally. You don't really need mutations for most of them, or they can be acquired through gene transfer, through plasmids, which we've documented again and again and again. There has always been some form of antibiotic resistance in bacterial populations in the world because, do you remember what the first antibiotic was? Penicillin. What's it come from? Mold. Molds grow in nature, right? Molds don't like bacteria. Molds have always had a way of protecting themselves against bacterial attack. So there have always been antibiotics in the natural world. We didn't invent them. So bacteria have always had some means available to them for antibiotic resistance. When we started mass producing them, then it just started spreading through all the bacterial populations. They started sharing and sharing alike. Okay? So nothing all that unusual biologically happened. And there was always a metabolic cost. For every one of these antibiotic-resistant adaptations for a bacterium, it always slows the bacterium down. It doesn't reproduce as fast. It doesn't metabolize as fast. It's a defective bacterium. They only survive in the presence of the antibiotic. You take the antibiotic away and put some wild-type bacteria in there with them, and they're going to disappear. The antibiotic-resistance ones will just disappear. They'll be outcompeted by the normal because the antibiotic's gone. So they're actually, antibiotic-resistance bacteria are technically defective bacteria. They're dealing with mutations. Antibiotic resistance does not involve the creation of any new or novel function in the cell. They exist naturally, and some form of resistance is therefore expected. There's no new genetic information required for antibiotic resistance, and that's one of the more famous examples used again and again of so-called evolution. Something else. I found this quote in a book from 1977, Pierre-Paul Grasset, a very well-renowned French evolutionist. His book was The Evolution of Living Organisms, but he didn't buy Darwinism. Didn't buy it for a second. He talked about bacteria here, and he said, what's the use of their unceasing mutations if they do not change? In some, the mutations of bacteria and viruses are merely the hereditary fluctuations around a median position. A swing to the right, a swing to the left, but no final evolutionary effect. He wrote a book on the evolution of organisms. And he's claiming here that mutations don't get you what you want. They don't accomplish what evolution truly needs. David Stern talks about one of the oldest problems, and this is in the journal Evolution 2000. One of the oldest problems remains largely unsolved, and he's pointing to exactly what we're talking about. Historically, the neo-Darwinian synthesizers stressed the predominance of micromutations, the kind of thing we were just talking about. Whereas others noted the similarities between some dramatic mutations and evolutionary transitions to argue for macromutations. So there's always been a bit of a dissonance. Is it accumulating micromutations, or do we need these macromutations to get real new novelty and adaptation? It's an ongoing controversy, 2000, even 2010. Sean Carroll, one of the major proponents now of Darwinian evolution, many popular books written now, And now, again, he says a long-standing issue in evolutionary biology is whether the processes observable in extant or real populations are sufficient to account for the larger scale changes evident over longer periods. Does microevolution just add up to macroevolution? In 2001, he's saying this is still unsolved. It's a long-standing controversy. 2002, a year later, Journal of Evolutionary Biology. Again, a persistent debate in evolutionary biology is one over the continuity of microevolution and macro, whether macroevolutionary trends are governed by the principles of micro. So here's our several leading scholars in evolutionary biology saying we've got a long-standing controversy. Do the small changes we see in antibiotic resistance, do they lead to the macro changes we might see in the Cambrian explosion? Scott Gilbert, a very well-known evolutionary developmental biologist. Therefore, he says, to study large changes in evolution, biologists needed to look for changes in now regulatory genes. The structural genes aren't good enough that make the embryo, not just the structural genes that provide fitness within populations. So the focus is shifting. Normal processes of evolution we've been studying now for 50, 60 years aren't telling us what we need to know. How do new organisms evolve? Where do they come from? What kind of processes do they require? He says, now we've got to start looking at these regulatory or these developmental genes. Another year later, Wallace Arthur. Textbooks of evolutionary biology have for years trotted out the usual old stories about how birds' beaks evolved to match their food items or how moths' colors evolved to match their background. You know what I'm talking about, right? But where are the equally detailed studies about the importance of one body part matching another? What he's saying is that the evolutionary studies we've done have only focused on too small a part of the organism. The beak of the finch is connected to the head. You can't change the beak without changing other things as well. If you're not going to study the whole head, then just studying the beak isn't telling you enough. Okay? Now, body plan is something we need to talk about. So we need to ask this question: what is a body plan? What I've pictured for you here are some of the arthropods, okay? Insects, crustaceans, centipedes, those kinds of things. They have a hard exoskeleton. They have numerous limbs. Some of them fly, some of them don't. Some are marine, some are airborne, some are on land. But then you have the chordates, which have a very different body plan. Their skeleton is internal, okay? And if they have appendages, they got 4, okay? Except for the fins of fish, they can have a few more, but you talk about mammals, birds, reptiles, there's 4 appendages, and that's pretty similar, unlike the arthropods. These are very different body plans, is how biologists talk about these body plans, okay? So the question is, how does the body plan form? Where does that come from? Now, structural genes tend to have little effect on the development of the body plan. You can mutate the structural genes and the body plan's not going to change. The developmental genes that do affect body plans tend to find their expression quite early in development, the 2, 4, 8, 32 cell stages. Those genes that control key early developmental processes are involved in the establishment of the basic body plan. So those are genes that control early development, okay? And they control the body plan. Mutations in these genes will usually be extremely disadvantageous, and it's conceivable that they are always so. Now that is a critical statement. If the body plans differ by these developmental genes that are active early in embryological development, and if you mutate those, they're always extremely disadvantageous. How then, by mutation, are you going to change from one body plan to another? You see the problem? Okay. The modern synthesis is a remarkable achievement, they say. However, starting in the 1970s, many biologists began questioning its adequacy in explaining evolution. Microevolution looks at adaptations that concern only the survival of the fittest, not the arrival of the fittest. I said back in 1984 that natural selection is a conservative process, not a creative one. It is about the survival of those that are already fit. It tells you nothing how the fit got there in the first place.